In the previous video, we distinguished between the digestive tract and digestive system. We also studied the digestion of proteins in the stomach by pepsin and in the small intestine by trypsin. Moreover, we indicated the first role of bile, which is rendering the medium basic in the small intestine. Recall that bile is produced in the liver, stored in the gallbladder, and conducted into the small intestine. Let's continue our journey in the small intestine in order to study the digestion of other types of food like lipids and maltose. Before we start with experiments, let's get to know the relationship between lipids and bile. So lipids are hydrophobic. Hydro is related to water, phobic means hate. In other words, there's no way lipids and water will mix. Why would that be a problem? Well, recall that digestion, also called hydrolysis, is a decomposition reaction in the presence of water. Since they don't mix with water, lipids will be hard to be hydrolyzed unless emulsified. What's emulsification? It is the process of dispersing the lipid molecules into the water molecules, so large oil drops will become small oil droplets mixed with water under the action of bile, making it easy for the enzyme to digest lipids. So in conclusion, the second role of bile is to emulsify lipids in order to facilitate their digestion by their enzyme. Now, after being emulsified by bile, let's study the digestion of lipids by their enzyme. In this experiment, the substrate is oil, which belongs to lipids, and the juice is the pancreatic juice. The adjacent graph shows the results of the experiment. Let's interpret the graph. At the beginning of the experiment, the level of oil was 100%, while that of fatty acids was 0%. The level of oil decreased to null, while that of fatty acids increased to 100% at time equal 180 minutes. This indicates that oil has transformed into fatty acids. And this leads us to the conclusion that the pancreatic extract contains an enzyme that hydrolyzes lipids. In fact, the pancreatic juice contains three enzymes: trypsin that acts on proteins, amylase that acts on starch, and lipase that acts on lipids. The functional diagram below shows the digestion of lipids by lipase in the presence of bile. After bile emulsifies the lipids, the lipase will break them down into fatty acids and glycerol. Note that each lipid molecule will be broken down into 3 fatty acids and 1 glycerol molecule. This is all. Hope you enjoyed the lesson. Study well and keep safe.